All right. Okay, so this week we're going to be covering the second half of chapter 7, which is specifically talking about how to control microbes in the environment. So last week we talked about how we want to promote their growth. This week we're going to talk about killing them. Okay, all right. So here is an overview of what we're going to cover today. We're going to go over the learning objectives. I'm going to open it up to questions. We're going to talk about legal definitions associated with products that are intended to control microbes in the environment. We're going to talk about this, what I call the scale of resistance, or groupings of microbes and how hard or how easy they are to control them in the environment. Then we're going to talk about some physical methods of control. Temperature, including heat and cold, radiation, filtration, pH, salt, okay? Interspersed will be practice questions, and then we'll go over what to do for next time. All right, so this chapter helps us move toward being able to meet the following objectives. Being able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. All right, any questions before we start? All right, if you think of some, let me know. I'm happy to answer them. Okay, first off, we're going to go to the whiteboard and we are going to talk about some legal definitions. Okay, we're going to start out with some definitions that are in your textbook, and all of these definitions are in Sarago. But I'm going to explain some of these definitions. Okay, so first off, sterilization. Okay, what is sterilization? We're all shy today.
That one's the one that kills all of the microbes, right?
Yep, kills or removes all microbes. Okay. Now we also toss in some non-living agents. Okay, so living things that are cellular, but we also want to include what kinds of things that are pathogens that aren't necessarily alive.
Repeat that.
So what are some things that are pathogens that are not alive that we would want to kill or remove as a part of sterilization?
Viruses.
Viruses, very good. Whoops, and I'm sadly misspelling that. Let me fix that. Okay, and is there anything else we would want to inactivate?
Endospores.
Yep, definitely. That would be including endospores. I'm going to put that down here. Including endospores. Okay. But we're going to toss in prions. Even though they are not alive, we still don't want them on stuff that is sterile. Okay. All right. This is the goal. Okay. Our goal is to kill or remove all living microbes, okay, viruses and prions. Okay. Now the problem is you cannot prove a negative. Okay, you can't prove that something is sterile. Okay? Here's an example. Let's say that I take 1,000, say, endospores. And I put it through a sterilization process and I get nothing out. Okay. Well, that sounds good. That looks like I've sterilized things. Okay. But there's always the question of, well, what if I put 10 times as many in? Okay. Am I going to get 0 then? Okay. And you can see that, we could keep moving the scale up, Well, if it takes out 1 million organisms, can it take out 10 million organisms? Okay. And so you never arrive at a positive, okay, we know it's sterile. Okay. Because you can't prove a negative. Now I can prove that if I put 1 million organisms on 10 million endospores, and consistently, after doing the procedure, okay, I only get 10 out, that I can prove. Okay, kind of see where I'm going, why you can't prove a negative. So instead, okay, because you can't prove a negative, what we've decided is that although this is a good goal, this is the goal that we have, okay, but we need to set benchmarks that manufacturers can reasonably prove, okay, when they're marketing a product that is intended to meet this goal. Okay, so sterilization is the goal, but a sterilizer or sterilant, okay, that is a product that or procedure that has met the benchmark. Yeah, that's spelling that wrong too. Okay, met the legal requirements. That's what I mean by benchmark. Okay, so in the United States, the FDA has said that something can be marketed as a sterilizer or a sterilant, okay, if they can show a 6 log 10 reduction of thermoresistant endospores. Okay, so what does that mean? And also it's greater than or equal to, okay? So what in the world does that mean? Okay, from last week, thermoresistant, okay? These are endospores that are formed by thermophiles. Okay, so we've got some bacteria that we isolated from a hot spring in Yellowstone. It lives at really high temperatures, okay, in the range of about, 100 degrees Celsius, okay, so it's at boiling, okay. Sometimes it's a little hotter, well, sometimes it's a little lower because, Yellowstone is at a fairly high elevation, but it's in approximately this range, okay. And endospores, when we talk about the scale of resistance, endospores are really hard to kill. So you take the really hard to kill ones and show that you can kill, okay, 1 million of these puppies. Okay? So that's what they do when they are meeting this legal requirement. They show that if they take 1 million of these thermo-resistant endospores, okay, and put them through their sterilization procedure or expose it to their sterilants, okay, that they can show that they have nothing at the end, okay. So what does this 6 log reduction mean? Well, log 10 means how many zeros, and this 6 refers to the number of zeros. 1, 2, 3, 4, 5, 6. So in other words, I've killed all 1,000,000 of them. Okay. Okay, how are we doing? Any questions? Okay, so hopefully that helps you understand what you're learning in Sarago. Remember, shun is the goal. Okay. Sterilizer or sterilant. Okay. That means that it met the legal requirement. Okay, so it's either equipment or it's a process.
Okay.
Yeah?
I have a question.
Sure.
So the benchmark is if they had 1,000,000 endospores, they'd be able to kill all of them.
Yep.
Okay.
Yep. So for example, we're going to be talking about autoclaves later on. Okay. But what we do to test them to make sure that they're functioning properly, we have a little vial, okay, that's a full of 1,000,000 of these endospores. Okay. So I've got a known number of endospores and they're floating around in media that we know that they're going to like. Okay. And usually endospores need some heat to get them to wake up and hatch out. Okay. And so what we do is we run them through the autoclave and then we incubate them for about 3 weeks. And if we don't get any color change in here indicating that the endospores woke up and started eating the media, then we know that there's 0 in there. Okay, questions? Okay, ready to go on to disinfection? Okay, so this is another one where we have the goal, okay? And then we have the legal definition of something that is supposed to achieve that goal, okay? So from Sarago, what is disinfection? What is the goal that we're trying to meet?
That's the reduction of the numbers of them, right?
Yeah, well, actually that is, let's go ahead and put that down here. Okay, so a disinfectant, okay, reduces, okay, by how much? Do you remember?
That one, 99%, isn't it?
99.9%. So you were really close. Yep. So
And that's
to be
the
marketed,
agent, right?
sorry, what?
So the disinfectant would be the agent.
Right. Right. So that is the agent. Okay. Okay. And so it reduces the test organism by 99.9%. So if we convert that to log reduction, okay, a disinfectant reduces a test organism by greater than or equal to 3 log 10. Okay. So what does that mean? That means that if and it doesn't matter what the test organism is, it doesn't have to be a thermoresistant endospore. It can be anything I want to test it against. Okay. So if I take 1,000,000, okay, let's say Staph aureus, because, you know, we're interested in preventing staph infections, okay, and I treat it to this disinfectant, okay, then I can show that consistently I get 1,000 or less out at the end of it, okay? So I've taken this number that has 6 zeros and I've knocked it down by 3 zeros. And that works out to be 99.9%. Okay. So that when you buy a disinfectant, they've proven that they can do this. Okay. But what's the goal? What are we trying to achieve? And in this case, the what they say you can meet to become a disinfectant is significantly less than what the definition is. And it's good to know that it's significantly different.
Wouldn't it be the same goal then as to kill them?
Yes, and so it is to kill. Okay, and because we're talking viruses, I'm going to put that in quotes because they're not alive. Kill or remove. Okay, all microbes, viruses. Okay, excepting endospores. Okay, that, in my opinion, is significantly different than what they require. Okay, what the EPA requires for folks who want to market something as a disinfectant. Okay, and that's good for you to know. Okay, now we have 3 levels of disinfectants. Okay, there are some, okay, that if I put in 1 million of a hard to kill organism, okay, and I can come out with 0. Okay, so let's say that I use an acid fast bacterium. Okay, and like the one that causes tuberculosis. Okay? And I subject it to my disinfectant and I consistently get nothing out. Okay, that has met the requirements of a disinfectant. Okay, because I didn't use thermoresistant endospores, I can't say it's a sterilant. Okay, but I can say that it is a high level disinfectant. Okay, now let's say that I barely made the grade. Okay, I put in 1 million. Okay, I have a relatively easy to kill microbe. Okay, let's say influenza virus. And now I say influenza virus because it's enveloped, so it's easy to kill. Okay, and let's say I barely squeaked through. Okay, and after applying it, I get 1,000 out. Okay, I have still met the requirements to call myself a disinfectant. Okay, but this is called a low level disinfectant. Okay, now does that mean that you should never buy a low level disinfectant? No, it depends upon what you're using it for. Okay, if I am trying to disinfect a surgical operatory, okay, you don't want to use a low level disinfectant. Okay, you want to use a high level disinfectant. If I'm wanting to disinfect a urgent care waiting room, okay, low level, 99% of the time is going to be okay. Okay. Yes, Rosa.
Hi, well, I just have a question. So you're saying the disinfection is kill or removal, but Cerebral says it's reduction.
Oh, okay. So yeah, I think we were going with Yeah, I tell you what, let's go with that one because I think we modified it to fit this textbook. So we'll put reduction in
It
there.
doesn't say kill.
Yeah, because you don't necessarily have to kill them. You just have to show that you've reduced the numbers, and it can be by kill or removal. Because both of those would.
Well, the reason
would-
I'm actually saying that is because, I'm sorry, because I actually put, there's one answer that is very close to reduction and the other one actually says kill. So when I actually did the kill, it was wrong. And the right one is actually the reduction of the numbers of bacteria, fungi, viruses, and other microorganisms. So it doesn't really say kill anywhere. Sorry.
Right. Yeah, because
I just want
remember
to
with,
make sure, right? Because I was, yeah.
well, the reason why we did that when we were setting up Cerigo is because we wanted to make this definition different from sterilization. So you are correct. In the definition in Cerigo, it doesn't talk anything about kill. Okay. But you can reduce the numbers by either killing them, okay, so that they don't grow, or in the case of viruses, inactivating. Or by removing. Okay.
But the legal definition is what it shows on Cerigo.
So in Cerigo, are you talking about disinfection versus
The
disinfectant?
definition, yeah, the definition of what shows there, you know, the reduction of viruses, but not the killing. The question is, like, if we get a question about this, the right answer will be reduction, or it will be okay to actually add language about killing or removing.
Yes, okay, yes, I see what you're saying, and I see what your concern is, okay?
Because I understand what you're saying, right? It can reduce, and based on the amount, then you know, it can kill, right, viruses.
Mhm.
But what I'm saying is, because you started saying about the legal definition, so we go by what is in Cerigo, or we go by, you know, this where yes, it reduces but also can kill.
Yes, so so yes, it's good to go with what's in Cerigo, but I'm trying to help you understand what it means in Cerigo. So if I change things up to show that you understand what you learned, as well as memorizing it, you're going to understand that if I say that I've killed, okay, 3 logs of Staph aureus, that's going to be the same as reducing the numbers. Does that help?
Yes. Thank you. Yeah.
Yeah, I know. I realize it's confusing. Okay.
Is the
But-
key between the two of them the endospores? Because in sterilization, you actually are going after the endospores, but in disinfection, you don't. You don't.
Right.
Correct.
Because endospores are hard to kill.
Right. So that would be the key between the two of them.
Yes.
Then would be the endospores.
Right. And we realize in disinfection, you know, because we're not getting rid of the endospores, okay, we would not really be able to say, you know, that we can say that there's a reduction, okay, but we haven't gotten rid of all the microbes because potentially we still have endospores. But in some textbooks, they say kill or removal of all microbes and viruses except for endospores. So my apologies on this one. I was going with a definition from an older textbook. So, but does that help that reduction, okay, can mean killing some of them, not all of them, okay? As long as I can show that I've that I've gotten rid of, I've knocked off 3 zeros from my starting, okay? That it could be killing, it could be inactivating, or it could be removing. And all of that will equal, okay, reducing the numbers. Does that help you move toward understanding?
Yeah, I think the one is correct. I think that I can understand better that the disinfection is not really, it doesn't kill endospores.
Right.
Yeah.
But we could remove endospores, but if we don't remove 6 log, then it's still disinfection. Does that help, or am I just confusing the issue further?
No, thank you. Yes, it does.
Okay. Yeah, and if I have not clarified things, I'm happy to explain it another way. You know, because I don't want to leave you confused. That's certainly not what I want to do. Okay, other questions?
You said there was 3 tiers, but you mentioned 2 of them.
Right, right. So I've got high level, which is pretty close to a sterilant. It's just like you said, there's no endospores involved. Okay. Low level is they're just barely making the 3 log reduction, 3 log 10 reduction. Okay. And then anything that's in between, okay, would be considered intermediate. Okay, so they were able to achieve a 3 log reduction of harder to kill organisms, okay? Bacteria, fungi, protozoa, okay? Harder to inactivate viruses, okay? So any of those would be intermediate level, okay? So we're looking at both the type of test organism and how much they were able to remove, to reduce. Okay. Okay, so as we go along this week, we're going to be talking about when it's good to use a low level, okay? Because there are times when a low level disinfectant is just fine, times when it's better to use an intermediate level, and times that you really should use a high level. Okay. Okay, questions? Okay, now one thing that I did not put in the definition up here is that this is when we're talking on non-living surfaces. Okay. So when I'm talking disinfection, okay, that's like countertops, pieces of equipment, okay? Let's move to what we're talking about, living tissue, okay? Antisepsis. Oh, and I've misspelled that. I think there's an E in there. Okay, so antisepsis, okay, is reduction, okay, of bacteria, viruses, okay, et cetera, okay, on living tissue. Okay, so that can be skin, okay, even though the outer layer is a dead layer of cells. After that dead layer of cells, you've got live cells underneath, continually replenishing it. We're talking mucosal membranes, okay? And it can, oh, go ahead.
So sterilization, is that for non-living too, or is it for both? Okay, okay, thank you.
Yeah. Yeah, because In the past, we've tried to sterilize, you know, skin without damaging the skin, and you know, it's just not possible. Because anytime you use a sterilant, you know, you're starting to go through the dead layer, and eventually it's going to get to your live cells, and we don't want to kill those. Yeah. So yeah, so we cannot, and we don't want to achieve 0%. Okay. Because we want to leave the living tissue untouched. Okay. And it depends upon what living tissue. We can use more harsh antiseptics on the skin, because the skin's pretty tough. Okay, we've got that layer of dead skin. Okay, so I can use things, and I can also use some disinfectants, you know, without wearing gloves, and it's not going to hurt me. Okay, but when we're talking the mucosa, and the mucosal membranes, the lining of the mouth, digestive tract, lungs, your eyes, okay? There, you know, it's a little more delicate. Okay? So in other words, you know, you don't want to gargle with a disinfectant, okay? I actually had a friend that tried a chemistry trick that involved taking Everclear, holding it in your mouth, and then spitting it into a sink where there was a particular chemical in the ethanol. And the chemical would react and they burst into flames, and it'll look really cool. And after doing that about 3 times, all of the lining of his mouth shed in one big clump, because it killed all of his mouth cells. So you want to be more careful with your mucous membranes, your mucosa. Okay, whereas you could get away with that with your skin and not be just fine. Okay, and generally that's why we do Everclear.
Was that a party trick or something he was doing?
Well, it sort of was a party trick, but it was actually in the stockroom of the lab, of the university lab where we were working. So anyway, I think he was working on a demonstration for a high school group that was coming in later, and he realized that's a once and done kind of trick. You shouldn't practice it too much in the same day.
Yeah.
So for antisepsis, we don't have a set benchmark that you need to meet. You just have to show that, depending upon the organism, that you can show that you have a reduction in numbers. Okay. Generally, like I said, for skin, we have antiseptics that are a little stronger. They have a higher reduction. Okay. And just because of how delicate the mucosa is, the mucosal membrane, okay, we generally see lower levels. Okay. So for example, okay, if I'm talking about a mouthwash, okay, say I've got some reddening of my gums, I want to prevent periodontal disease, okay, I'm going to use a mouthwash, but it's going to be more gentle. It's going to reduce the numbers less, okay, then say I'm going in for surgery and we paint iodine. Okay, iodine containing antiseptic on the side of the surgery, okay, because we want very few things to get into that surgical site. Okay, having surgery is already an insult to the system, and your immune system is going to have to go in and take out skin bacteria and whatever has gotten into your tissues that shouldn't get into your tissue. And we want to keep those numbers down. Okay, and you can do that to your skin and not have as big a problem as if you would want to do the same thing over here. Okay? Now we do have mouthwashes that are that have iodine in them, but it's a lower level of iodine. Okay, questions about antisepsis and how that relates to antiseptics.
Does the book go into dwell times of these agents?
Yes, yes, and which is dwell time is an important thing, okay? Because iodine has a longer dwell time. Okay, it's not going to evaporate off, okay? Whereas if I'm talking about, say, Purell, and I use Purell because it's a brand name that I think most people are familiar with, and its main ingredient is alcohol. Okay, it has a shorter dwell time. Okay, so in other words, it's going to stay on the skin for a shorter amount of time because that alcohol is going to evaporate off. And so alcohol is not going to reduce the numbers of bacteria or other microbes on your skin as well as iodine. But it's less expensive to use alcohol. And also for most of us, because, you know, we're not going to have surgery done on, you know, our hands, unless, you know, you're going in for surgery and then they're going to paint iodine containing antiseptics on your skin. You know, you don't need it. You don't need it. Did that answer your question?
Yeah, but also the dwell time also depends on if those agents actually are going to work properly. Because if you don't let them dwell long enough, then they don't actually perform and kill as much of the bacteria as it's supposed to, because you didn't let it dwell as long as it's supposed to.
Yep.
Correct?
Yep, that is correct. And that is correct when we're talking about sterilizers and disinfectants. Okay, because dwell time, we also call it contact time. Okay, those are synonyms. Okay, it's how long that you have the chemical, and particularly when we're talking chemicals, how long you have that in contact with what you're trying to control the microbes on. Also, it's okay, I have a thought and it just left my brain. For sterilizers, it's how long, say with an autoclave, it's how long you run it. Yeah. So yes, contact time, dwell time are both very, very important. Yeah. And while we're talking about it, let's introduce the topic of bioburden. Because contact time, dwell time, okay, the active ingredient, and also bioburden are all important. Okay, actually, I'm going to have to clear the board because I'm run out of room. Okay, so let's say, let's use an example that we're familiar with. Okay, let's talk about HIV. Okay, HIV is an enveloped virus. I'm going to go ahead and put some short little spikes on here. Okay. Now, if I have HIV in a test tube in a lab and it's just floating around in, say, physiological saline solution, okay, so a salt solution that's at, if I, well, it's not rounded, 0.09%, okay. So it's a physiological saline that's going to keep that alive, okay. But if I say I stick this in, okay, say, you know, an autoclave, okay, I'm going to be able to activate that virus very quickly, okay, because autoclaves work by heat, and heat's going to be able to get in. There's not much blocking the heat, okay, from killing the viruses, okay. Now, let's say instead I have the HIV, okay, and instead of being in a salt solution where I've just got salt and water, okay, and not even that much salt, okay? Let's say I've got a vial of patient blood, okay? So I have, and I'm not going to draw them in their correct filled in lifesaver shape. But let's say I've got all sorts of red blood cells in here, okay? And I'm not even packing them as close as they would be, okay? And this is from a patient sample, and I have all sorts of HIV. This is from a patient that is HIV positive, okay? So they have virus in their blood. And so, well, they're probably not going to be inside of the red blood cells, but they would be inside of some of the white blood cells. Yeah, so let's go ahead and get some white blood cells in there. Okay, so if I autoclave it, okay, the heat has to get through the blood cells and the blood protein, okay, to get to the virus. Okay, so we call that bioburden. Any biological substance, okay, that insulates, if you want to think of it that way, okay, the pathogen that we're trying to kill. Okay, so I've used blood as an example of bioburden. Okay, what's another example of bioburden? What about with COVID?
Mucus.
Mucus. Okay, so that protects the COVID virus, the virus that causes COVID, okay, prevents it from drying out in the environment. And then if I'm trying to disinfect, okay, the disinfectant, okay, or the sterilant has got to get through the mucus to get it to the virus, okay? What's another one? What's another bodily product that could insulate, that could protect unintentionally a pathogen?
Does it have to be a fluid?
Nope, it doesn't have to be a fluid.
Could the lipid bilayer protect?
Yes, yes, especially if it's a lipid bilayer of a host cell. Okay. But when we're talking about controlling the environment, lipid bilayers are kind of hard to see. So we generally talk about stuff that you can see. Yes, Courtney.
Tissue.
Yep, tissue. Okay, so if I have taken a biopsy, okay, and I need to dispose of that biopsy, but I want to make sure it's sterile first, okay, the tissue of the biopsy is definitely going to be bioburden. Okay. What if I have somebody with a stomach complaint? What are some of the symptoms of a stomach complaint that could be considered bioburden?
Diarrhea.
Diarrhea. Okay, slash feces of any kind. Oops, that's a C. Okay, what about the other end?
Vomit?
Vomit. Okay, vomit is definitely considered a bioburden. Okay. Also saliva. Okay. Urine. Okay, anything that comes out of a body, okay, would be considered bioburden. Okay, so when you have more bioburden, you need to have a longer dwell time. Yeah. Or if you're doing a physical method of control, you need to do it for longer because you have to get through that bioburden to kill the pathogen. Okay, kill or reduce the numbers of the pathogen. Okay. And next time when we get to sterile or chemical methods of control, okay, the difference between a chemical sterilizer and a chemical disinfectant, okay, is often a difference of time, okay, and making sure that you do it long enough to get through the bioburden, okay. So for example, okay, a 1.5 dilution of bleach, okay, if I leave something in it for 8 hours, okay, it'll sterilize it, take out those endospores. Okay. If I do it for 30 minutes, okay, it's a disinfectant. Okay. So that goes back to your statement, Devon, about how dwell time is important. Okay. And this may be longer if I'm trying to disinfect a clean surface, okay, no visible bioburden, versus trying to disinfect a dirty, okay, something with visible bioburden, okay, you'd have to do it for longer. Okay, questions? Okay, before I ask you questions, we're going to hit one more term. Okay, sanitization. Okay, so once again, this is the goal. Okay, so what is the definition from Sarago? Okay, it is, we've got reduction again. Reduction of the numbers, we're going to go with the abbreviation for numbers, of reduction of the numbers of potential pathogens to a level that should not cause disease. Okay. Now, personally, this definition confused me for years, okay? Because it means something different in a home versus when I was working in food service, working in restaurants and what have you, versus when I worked in clinical environments. Okay, because we're working with different potential pathogens and we're talking about different numbers, okay, and we're talking about different people's immune status. Okay, now in a home, okay, you know everybody who lives there. Okay, so we're talking family. Okay, can also be friends. Okay, but they're people that you have close contact with on a regular basis. Okay, and usually, okay, it's a fairly small number. Okay. Okay. And you know everybody's immune status. Okay, you know if somebody in your family is undergoing chemotherapy for cancer and they're immunosuppressed. or if they just had a kidney transplant and they're on immunosuppressive drugs to prevent rejection of the kidney. Okay. So generally, the need for controlling microbes varies depending upon the home. Okay. So it's going to be different for everybody's home. Okay. But unless you have somebody who has a compromised immune system, generally your need to control microbes in your home is what I call episodic. Okay. Okay. Little Timmy comes home from daycare and starts vomiting and has severe diarrhea, and it goes through the whole family, and then it's over. Somebody comes home, they bring a cold, everybody gets it, and it's over. Okay. So it's brief episodes. Okay. And then, like I said, unless you got somebody who has a suppressed immune system, okay, generally speaking, you don't have to have stringent requirements to meet sanitization, to reduce the number of potential pathogens to the level where it's not going to cause disease. Okay, where the vomiting and diarrhea is going to continue to cycle back and forth. Okay. Now you get over to food service, okay, and you have larger numbers. Okay. Of people. Okay. And except for the staff and regulars who come in on a regular basis, okay, they are all strangers. Okay. So you need to up your game a bit. Okay. But generally speaking, okay, most of the people that go out to eat, okay, are healthy. Okay. Unless you're doing Meals on Wheels, you know, then you're taking food to potentially folks who are, you know, homebound, who may have more of a compromised immune system. Okay. So it depends upon your food service, but generally speaking, we're talking about healthy people who work there. Okay. If you work in food service and you don't feel good, do not go into work, especially if you have vomiting and diarrhea. Just don't do it. Okay. Okay, so you're generally healthy. Okay, so the numbers are going to be higher. Okay, for which should, emphasis on should, cause disease. Okay, and we're dealing with food and waterborne pathogens. Okay. Those are the main ones you're concerned about. So it's a select number or a select type of pathogen. Okay. So sanitization is going to mean something different than it does at home. Okay. One, because of the volume of food you're making and the number of people that you're providing services to and that kind of thing. Okay. Now with clinical, okay, you've got the large numbers of people. Okay, and all of them are there for a reason. Okay, if you're running a standard general practice doctor's office, you may get a lot of people who are healthy and they're just there for their yearly checkup. Okay, but in my experience, if you're healthy, you tend to forget your yearly checkups. So generally, you have large numbers of sick people. Okay, sick or injured. So we're talking folks who are bringing in large numbers of pathogens, all sorts of different kinds of pathogens, and we have large numbers of potentially immunocompromised. Okay, because they're sick. Okay, when you're injured, that slows down your immune system because it's busy, you know, trying to heal your injury. Okay. So what sanitization means in the clinical environment is different than in food service than in home. Okay. So generally, when we talk about sanitization in clinical environments, Okay, we are generally talking intermediate to high level disinfection. Okay, in a clinical environment, sanitization is synonymous with that. Okay. When you're talking food service, we're talking low to intermediate. Okay. And in the home, depending upon what your home situation is, generally it's low. Okay, because the needs are different, the goals are different. Okay. Now, having said that, if you want to market something as a sanitizer, okay, it is a failed disinfectant. Okay, it shows, okay. Oh, by the way, this is all non-living. Okay.
Can you repeat what you just said about the sanitizer?
Yes, sorry, I just put non-living. Okay, let's try this again, and then I'll repeat what I said before. Non-living. Okay, so a sanitizer is a failed disinfectant. Okay, it didn't meet the benchmark, the requirements for reduction of a pathogen, a non-endospore pathogen. Okay, so it did not make the 3-log reduction. Okay, so if you can show less than 3-log reduction, but you're still showing a reduction of a test organism, you can market yourself as a sanitizer. And you know what? Most sanitizers are really good cleaners. So that is not to poo-poo sanitizers. Sanitizers do a really good job of cleaning. Okay, and you want to get rid of bioburden. Okay, just realize that this is what sanitizers do. Okay. Okay, questions?
I have one question. So
Sure.
for sanitizer, it would be 1,000 would be a disinfectant, and so would the number be over 1,000 if it was a sanitizer for living?
So here's an example. Let's say that I took, and we generally always go with 1 million. Okay, so let's say we're looking at 1 million staph aureus. You know, because we're interested in preventing staph infections. If it is a sanitizer, okay, I would show, yes, a higher number out, less of a reduction. Okay, so let's say instead of getting 1,000 out, I got 10,000. Okay, that would be a 2-log 10 reduction because I knocked off 2 zeros. Okay, that's not to the 3-log reduction. Okay, so then I would say that I'm a sanitizer.
Okay.
So sanitizer is less than 3-log 10.
Yes. Yep. Yes. Less than 3-log 10 reduction. So I did not succeed in knocking off 3 zeros. Any other questions?
I have a question. So it just has to do with the zeros then. So if it knocked off, like if you put in the 1 million and then it came out to like 9,000, then it would meet the, okay, thank you.
Yeah, and I generally deal with whole numbers like this. Well, base 10 numbers, just because it's a little easier to figure out. Because like if I had a 3-log reduction, I just X out 3 of these zeros and I've got 1,000. Actually, if I went 2.5-log reduction, it'd be 5,000. And on the test, I would do, you know, where you can just knock off the zeros.
Okay, thank you.
Quite welcome. All right, so let's see, we have about 15 minutes left. Tell you what, instead of asking you questions, let's go on to some of the physical methods of control. Okay. But I am in the process of putting the questions from the PowerPoint into the chapter 7, part 2 practice quiz. So you can practice with those questions, and I'll also put in some more application style questions that you can work on. Okay. So I'm expanding the pool.
Thank you.
Quite welcome. Because, you know, there's never enough time in lecture. Okay, let's talk about temperature. Okay, temperature is a physical method of controlling microbes in the environment. Okay, so here's why. Let's go ahead, and this goes back to what we've talked about before. Okay, especially when we were talking about gross conditions. Okay, so this goes with the previous part of the chapter. If I heat things up, the molecules are moving around, they're bouncing into each other. Water is bouncing into the proteins are bouncing into the phospholipid bilayer. The phospholipid bilayer molecules are moving around. Okay, so when we talk about proteins, okay, because proteins, especially enzymes, tend to be very sensitive, okay, to differences in heat. Okay, let's say that this part of the functional structure is held together with, say, an ionic interaction here, okay? And then I have a hydrogen bond here, so I've got some OHs that are holding hands, okay, across the way here, okay? If these move around too much, okay, this gets moving this direction, this gets moving this direction, and these are too far away to interact, to have an attraction, okay? If this moves far enough away, these are too far away to interact, and I break the hydrogen bond. Okay. What do I call that when I unravel a protein from its shape? I haven't broken the covalent bonds that hold the individual amino acids together, but I call that what?
Denature.
Very good, denature. Okay. So heat will denature proteins, okay? They will also disrupt lipid membranes. You get those lipid, the phospholipids in the membrane bouncing around and moving too much, and they're basically, the cell's gonna pop, okay? Nucleic acids are sensitive to heat, okay? For the same thing, you start pulling them apart, you break those hydrogen bonds between the bases, okay? You want it to happen controlled through enzymes when you're replicating the DNA, and when you're transcribing the DNA to make proteins, but you don't want to do it randomly because of outside temperature, okay? So heat will kill, okay? If we're talking cellular organisms, if we're talking viruses, it will inactivate. Okay, because viruses aren't really alive, so we can't really say that we're killing them, okay? So heat will do that. And we have two different types of heat that we routinely use, okay? So in the lab, we use a fancy dancy thing that we call an autoclave. And the principle of an autoclave is the same as for a pressure cooker. Okay? Except for we tend to clean our Instapots better than we clean autoclaves because we're not trying to have anything be delicious after we get done. Okay. So basically with both of them, okay, we're increasing the pressure so that we can get a higher temperature. Okay. And they are both moist heat. Okay. So we're using water, okay, we're using pressure to increase the temperature, okay? And so because we're able to get a higher temperature, okay, we're able to have the heat go through bioburden better. Okay. So both of these are examples of what we call moist heat. Okay. Now, let's see. Yep. So, okay. Now we also in the lab use what is called dry heat. Okay. So with dry heat, okay, when you're doing this at home, your oven is considered dry heat. Okay. You can sterilize stuff in your oven. Okay. When we cook stuff, we're generally not going for sterilization because, you know, we like our food to still have some water in it instead of being dry and crispy. Okay. So an oven though is considered dry heat. Okay. We have to use it for longer because we're not using water. Water transfers heat better because the water molecules are helping to bounce into the proteins we're trying to disrupt, that we're trying to denature. Okay. So we've got some more spread out air molecules, so you've got to heat it up hotter, okay, and for longer. Okay. And then also in lab, we use incineration. Okay, those Bacto incinerators. Back to there we go. Okay, and also, you know, if we weren't using Bacto incinerators, we would use a flame from a Bunsen burner. Okay. Now with the Bacto incinerator and with flames, what we're doing, okay, not only are we denaturing the protein, we're also breaking the covalent bonds between the amino acids. Okay. And then we continue to break those down. We break down the amino acids. Okay. So I'm taking, oops, I left my nitrogen off. Okay. I'm taking something that looks like this, and this is very abbreviated form. And we're turning it basically into carbon dioxide. Okay. So we're breaking that down completely as small as we can get it. Okay, and so that is incineration. And that is a dry heat method of control, okay, that, you know, that we use frequently in the lab. It's a good way to sterilize, and it's pretty quick. Okay.
Would a dehydrator be dry heat also?
A dehydrator, actually, even though we're using heat, we put that into drying, which is part of osmotic pressure. Okay. So we're removing water, we're increasing the salt concentration, and that will also denature proteins. Yeah, so you can kind of consider it temperature, but you can also dehydrate cold. Yeah, sometimes we do some mix and match. Yeah. Okay, let's go ahead and put a dividing line between my heat and my cold. Whoops, that was a terrible dividing line. Went right through the incineration. Okay, so for cold, we're doing the exact opposite. Instead of moving the molecules around more and faster, we're slowing them down. And what happens is my proteins, especially my enzymes, here's my cute little cartoon of an enzyme. Okay, here's my active site. They move slower and slower until finally they're hardly working at all. Okay. Now, when we're talking microbes, okay, oops. We've got 5 minutes left. I'm gonna have to, I want to talk about filtration real quick. And then also you'll notice this week I have recorded lectures that I want you to watch. So even if I'm not able to get everything today, we do have some recorded lectures that go over all of the stuff that I didn't have time to get to. Okay, so microbes have adapted to go into dormancy. Okay, so they basically go into hibernation. Okay, so they go to sleep for the winter and they wait for things to warm up. Okay, and so we use cold to preserve. Okay, rather than kill or inactivate microbes. So we use cold all the time. So when people say, oh, you can stick it in the freezer to kill anything that's in there, yeah, that's probably not going to work. I would not suggest that. Okay, it might kill things like insects. I'm not an entomologist. I just heard from a friend that if you want to preserve flour and prevent it from getting weevily, that you can stick it in the freezer and give it a good hard freeze, and then keep it in the sealed bag and then stick it in at room temperature and it won't get wiggly. And I'm going, you know what? I know of a lot of bugs that can survive the winter as eggs. So I'm not sure that'll work either. Okay. Now, the reason why we tend to think that cold will kill, okay, is because cold will kill humans. Okay, we humans have not adapted to surviving, hibernation. We don't go into hibernation when we get cold. We get into hypothermia and we die. Okay. Now that's most of the time. There's always exceptions. Okay. But we humans, especially our brains, have to keep at a certain temperature to keep functioning. Okay. When your brain goes into dormancy, it's not too much longer before the cells start dying. Okay. Our brains are very, very finicky that way. They're very touchy about the temperature. And that's why we have evolved to keep our body temperatures, our core temperatures, at a certain temperature. Okay. Now, when we're talking organ donations, you can keep them refrigerated for about 3 days, and then they start to die. So things like livers and kidneys and that kind of thing, hearts, are a little bit more resistant to colder temperatures. Okay. But that's why people tend to think, oh, if I put something in the freezer, it's going to kill it. Okay. That applies to people. That applies to mammals that have not evolved to go into hibernation. You put a bear in the freezer and it's going to be just fine if you give it enough time to get ready. Okay. But with bacteria, you freeze them down slowly and they do just fine. Okay. All right. We have 2 minutes left. Okay, filtration. Let's do filtration real quick. Okay, so the principle of a filter is to remove. Okay, a filter is not going to kill or inactivate, but it will remove. Okay, and we use filters, okay, for air and some fluids. Okay, so if I'm talking water, okay, and I want to get rid of bacteria in water, okay, let's say I want to get rid of intestinal bacteria. Intestinal bacteria are huge. They're going to get stuck on the filter. Meanwhile, the water is going to work its way through the holes and come through with fewer microbes. Okay. Now, sometimes we have itty bitty little microbes that can work their way through, but those are fairly rare. Okay. Same thing with air. Okay. Most viruses, if they were floating around free, okay, on the air, okay, are going to make it through a filter just fine. Okay. But generally speaking, viruses are riding on globulates of mucus. Okay. Mucus is going to get stuck, and the viruses with the mucus are going to get stuck. Okay. Little dust particles. Okay. Filters will catch a lot of dust particles. Some of them are going to get through and they're going to carry the viruses and what have you. But a lot of dust particles are even bigger, okay, than bacteria. Okay, so the principle that we want, what we're trying to do with filters, is physical removal. And by physically removing, okay, you can sterilize. Okay, you can physically remove greater than or equal to 6 logs. Okay, so if I take 1 million endospores, I can show that they all got caught here and didn't make it through to the other side. Okay, we can also disinfect. Okay, and it all depends upon how big the holes in your filter are. And say in masks, how many gaps around the outside you got? Okay, questions about filtration? Okay, my apologies. I have gone over time, but let's go to what to do for next time. Yeah. Go through all this fun stuff. Oops, I went too far. All right, so make sure you go through the contents of the week 9 lecture folder. Okay. The embedded tutor assignment number 2 is due this Friday. Okay. And go ahead and start working on case study number 3. It's not due until after spring break. And my goal is to have case study number 2 hopefully done before spring break, but we'll see how well I do. Okay, any questions?
Are our tests open now so we can go in and look at our results?
Yes. You
'Cause I
should
tried
be
it
able,
earlier and I couldn't get in.
I also tried.
give it a try now.